OsteoBites. Happy Thursday, and thanks for taking time out of your day to join us. My name is Christina Iptoma, and I am mom to Osteo Angel Dillon and Director of Scientific Programs at MIB Agents. And today on OsteoBites, we are joined by Dr. Sabina Kaczanowska from the Pediatric Oncology Branch at the National Cancer Institute, and she will discuss immune determinants of CAR-T cell expansion in solid tumor patients receiving GD2 CAR-T cell therapy. Thanks so much, Dr. Kaczanowska, for joining us on OsteoBites today. We are thrilled to have you. And thank you also to our panelist, Andrew, for joining us. Everyone, please feel free to submit questions during the presentation through the Q&A in Zoom, and we will be sure to share those with Dr. Kaczanowska. And first, a little bit more about our guest today. Dr. Sabina Kaczanowska received her PhD in molecular microbiology and immunology from the University of Maryland, Baltimore for her dissertation work on T-cell immunotherapy in the Marlene and Stuart Greenbaum Comprehensive Cancer Center. She joined the tumor microenvironment and metastasis section of Pediatric Oncology Branch at the National Cancer Institute as a postdoctoral fellow in 2017 with Dr. Rosandra Kaplan, who also happens to be on our MIB Scientific Advisory Board. And Dr. Kaczanowska's research focus is to understand the role of the immune system in shaping the microenvironment of metastasis during cancer progression. Specifically, she's investigating how myeloid cells orchestrate the immunosuppressive program in cancer and how we can apply this knowledge of myeloid biology to design new immunotherapeutic strategies for the treatment of solid tumors. Dr. Kaczanowska was appointed as a staff scientist in the Pediatric Oncology Branch in 2022 to continue her work evaluating the responses of solid tumor patients to immunotherapy and translating new immuno-oncology approaches into the clinic. Thank you so much for joining us today, Dr. Kaczanowska. We've been excited to talk to you since your paper was published, I think, maybe in January of earlier this year. So we've been waiting a while, so we're super excited to have you. Just a couple of quick announcements before we get started. As we bridge July Sarcoma Awareness Month and September Childhood Cancer Awareness Month, we are all about the patient experience informing research and clinical best practices, and there are a couple of ways that patients and families and even clinicians can contribute info and data. First, patient families are invited to participate in a survey to better understand the global reality of sarcoma patients' diagnostic experiences. Delays in diagnosing sarcomas are really common, and there's not much data or published information on sarcoma diagnosis. So the studies we have are often just focused on a specific region or a specific subtype. And so it leaves us with some gaps at our global level of understanding. So our advocacy partners at the Sarcoma Patient Advocacy Global Network are spearheading an effort to fill this void. And so they've launched a survey. You can share your experiences about your sarcoma diagnosis from initial symptoms to post-diagnosis management, and it will really help inform our understanding of diagnosis around the world. So I'm going to put a link to that survey in the chat. And then there's actually a second effort that I wanted to mention as well. It's CURE-ID, which is an initiative with the FDA and also the NIH National Center for Advancing Translational Sciences, also known as NCATS. It's a website and also a mobile app, and it enables healthcare providers, patients, and care partners to share real-world treatment experiences with existing medications that are being used in a new way through a simple online case report form. So you can enter it on your smartphone or on your computer. And CURE-ID then captures treatment outcomes when medications are used to treat new diseases beyond the conditions that they were originally studied for. So basically drug repurposing, or you might have heard the term off-label drug use. Healthcare providers, patients, and care partners can submit their case reports to help identify promising new uses of medications. And so this systemic collection of real-world treatment experiences reported to CURE-ID can serve as a resource for the community to share information where there is no FDA-approved product that is proven safe and effective. So that's really the case in osteosarcoma. And all those cases are made openly accessible. So this is right in the beginning phase. And so in addition to submitting case forms, I know they would also appreciate feedback as they grow the program. I think as of now, there are 4 osteosarcoma cases in there. And you can actually check them out. And even though it's not a lot of volume, it's just kind of cool because you can just see how the information is presented. So I'll put some more information in the chat about that as well. Andrew, could you go ahead and introduce yourself, please?
Hi, my name is Andrew Basaga. I was diagnosed in 2017 with osteosarcoma in my proximal femur, but I have now about 6 or it might be 7 years on NED now. And I am now a rising sophomore at Creighton University.
Awesome. Thanks, Andrew. All right, Dr. Kaczanowska, if you want to go ahead and start sharing your slides, that'd be great.
Yes, thank you. Can you see my screen?
Yes, that looks perfect.
Perfect. Thank you so much for the introduction and for the opportunity to present for you today. It's really an honor to represent this work. And this really is the epitome of team science. I'm the one here presenting this, but this was the effort of many, many people over a decade of work to put this trial together, put these correlative studies together. So I hope I can really do that justice. As Christina mentioned, I'm a staff scientist with Dr. Rosie Kaplan in the pediatric oncology branch. And today the project I'm going to be talking about is mainly focused on correlative studies from a phase 1 clinical trial looking at GD2 CAR-T cells in solid tumor pediatric patients, mainly osteosarcoma and also a few neuroblastoma. So this was a large multi-institutional collaborative effort to put this together. This trial was initially started by Crystal Makel, who was at the time chief of the pediatric oncology branch at the National Cancer Institute in Bethesda, Maryland. After Crystal left to go to Stanford, my PI, Rosie Kaplan, took over the trial and seeing the patients on this trial. So here's the clinical trial information. This trial enrolled between 2014 and 2016. I joined the lab in 2017 and was tasked with trying to organize all the patient samples and try to get everybody together to learn from these patients and learn from this trial. So myself and a graduate student in the lab, Chrissy Contreras, led the efforts on the NCI side. And this work was really spearheaded by Sneha Ramakrishna, who now has her own group at Stanford. She trained under Crystal, and she is continuing to do similar correlative efforts in ongoing trials looking at GD2 CAR-T cells, both in peripheral solid tumors as well as in pediatric brain tumors. And our analysis kind of focused on the immune system and mainly on the T cell compartment and the myeloid cell compartment. So Stanford, the team at Stanford really took the point for analyzing the T cells from these patients. And a lot of that work was done by Tara Murdy, who's a graduate student in Crystal Makel's lab. And then the myeloid focused portion of the analysis, we recruited our collaborators from the La Jolla Institute for Immunology, Catherine Lynn Hedrick, who is an expert in monocyte biology, and her scientist, Ahmed Alamadadi. And their group has since moved to Augusta University in Georgia, but we're continuing those collaborative efforts as well. And then the last kind of group that was involved heavily in this analysis was Jen Altruer's group at the Dana-Farber Cancer Institute, and they were really instrumental in analyzing the RNA sequencing data from these patients. So just to kind of start a little more basic, we know that our immune system can recognize and respond to cancer. And you think of your immune system as being a barrier or like a protective force to help protect you from outside pathogens like bacteria and viruses and help clear infections. But it is now very well accepted that the immune system can also recognize your cancer cells, because although they arise from normal cells in your body, they do acquire mutations and can appear different than self to your immune system, and your immune system can recognize that and can actually kill the tumor cells. And there are many different types of immune cells, and you'll hear me kind of mentioning some of these throughout the talk. But one of the cell types that people have really focused on are T cells, and this is for their ability to recognize cancer cells and to even directly kill them. So the CAR-T cell therapy that I'll be talking about is built on these T cells. But it's important just to note that all of your immune cells can be changed by cancer, both locally at the tumor site and also systemically. So all of the samples that we analyzed for this study were in the periphery, so circulating blood cells. And the idea behind GD2 CAR-T cells, so it's a chimeric antigen receptor, meaning it's a synthetic receptor that's designed to help the T cell better target the tumor. So in this case, and I'll talk about it a little later in the next slide as well, but we're targeting an antigen that's expressed on the surface of osteosarcoma and some other tumors called GD2. So the tumor recognition portion of the chimeric antigen receptor is based on an antibody fragment that recognizes this molecule that's found on the surface of tumor cells. And then the internal portions of the CAR act to initiate signaling into the T cell to activate that T cell to then kill the tumor. So kind of a basic mechanism is if you have your CAR-T cell, it will recognize this tumor-associated antigen, in this case GD2, on the surface of a tumor cell, and it can kill this tumor cell through a variety of mechanisms, including granzyme and perforin, which kind of punch holes in the tumor cell and cause it to lead to apoptosis or necrosis. And CAR-T cells can also secrete cytokines that can recruit and activate other immune cells, such as NK cells. So the idea behind CAR-T cell therapy is that this is a personalized or autologous therapy where cells are apheresed from the patient. They're genetically engineered in the lab. So they're used a virus to introduce this chimeric antigen receptor that's then expressed on their surface. These cells are activated and expanded in culture, and then they're infused back into that patient. And the idea is that now you've equipped these T cells to both recognize the cancer cell and receive activating signals when they encounter the cancer cell to hopefully attack and kill that cancer cell. So the target for this particular CAR-T cell is GD2, which is a ganglioside that's highly expressed on the tumors, but importantly has very limited expression on normal tissues because you don't want these highly active T cells to be targeting your normal tissues. It's expressed highly in osteosarcoma and neuroblastoma, as well as some other pediatric solid tumors. So here in A, you can see a metastatic osteosarcoma tissue section. This dark brown staining indicates presence of the GD2 molecule that's very highly expressed in the metastatic tumor here, but it's not expressed in the adjacent normal lung here. And also here it's shown that's highly expressed in rhabdomyosarcoma. And on the bottom here, just looking at expression by flow cytometry on numerous different sarcoma cell lines showing its expression. So it is highly expressed. And actually a monoclonal antibody that targets GD2 is part of the therapeutic backbone for the treatment of neuroblastoma. And groups have previously performed trials studying GD2 CAR-T cells and their effect in neuroblastoma patients, but this was the first trial looking at the efficacy of these CAR-T cells in osteosarcoma.
Dr. Kaczanowska,
So, yes.
Sorry, quick question on that. Do all osteosarcomas express GD2 and what is the range of expression?
For the most part, I believe the answer is yes. I'm not a clinician, but I think osteosarcoma is one of the ones that I think the vast majority of patients do express it at high levels. Some of the other solid tumors, I think there's a little more variability, but it is highly expressed in osteo.
Thanks.
So the trial performed at the NCI was a phase 1 clinical trial, and the primary objectives were to determine the feasibility of producing these CAR-T cells and then the safety of administering these CAR-T cells. They were given with a lymphodepletion regimen that consisted of cyclophosphamide given a few days prior to the CAR-T cell infusion. And then the CAR-T cells were given at escalating doses. So we had 4 dose levels here going up to 10 million transduced T cells per kg. So the patients would come in, they got apheresis. So this is to collect their T cells in order to manufacture the CAR-T cell product. During this time, they received their lymphodepleting chemotherapy and then were infused with their CAR-T cells on day 0. We have 28-day cycles where the patients were monitored. And then we also had some samples for long-term follow-up. And all these patients just received a single dose of the CAR-T cell therapy. So some of the samples that we were looking at for our correlative analysis was baseline samples. So these are apheresis or peripheral blood samples that were taken prior to treatment with the CAR-T cells. We were able to analyze the CAR-T cell product itself. So this was what was manufactured and given to the patients. And then we had some limited post-treatment samples as well. So the primary objectives for this trial were met. We were able to successfully manufacture GD2 CAR-T cell product for all the patients, and administration at all 4 dose levels was safe. So only 2 out of the 13 patients that were treated had grade 1 CRS, and there were no neurological or dose-limiting toxicities observed on this trial. And this is a swimmer's plot showing the clinical results from this trial. So 15 patients were enrolled, 13 of which were with osteosarcoma. 2 patients did not receive cells, and the remaining 13 patients were successfully treated with the GD2 CAR-T cell therapy. And you can see that some patients had stable disease, but unfortunately, eventually all patients did progress or switch to alternative therapies. And although we didn't really have clear responders or non-responders clinically on this trial, one thing that we did note was that there was a large variability in the expansion of the CAR-T cells after infusion into the patients. So we saw that some patients had a very robust expansion of the CAR-T cells, whereas some had little to no expansion of the CAR-T cells. So we decided to focus our correlative analysis on stratifying the patients based on this parameter. So here you can see how we grouped our patients either into the good expander bucket, which had high GD2 CAR-T cell expansion, or the poor CAR-T cell expanders, which had little to no expansion. I just want to point out that these patients that had good CAR-T cell expansion, this expansion was to a similar level that we see in, for example, CD19 CAR-T cells, which are effective in leukemias and lymphomas, whereas this was not enough in itself to have clinical response in osteosarcoma. And also that these, even though some of the patients had really good CAR-T cell expansion, they didn't really have persistence in these patients. So this is a schema of the different analyses that we were able to do with these samples. So first we wanted to look at the T cells, both in the baseline apheresis and the product. And this T cell phenotyping was performed at Stanford. We also wanted to look at myeloid populations at baseline prior to CAR-T cell treatment. And this analysis was done at the La Jolla Institute for Immunology. And then we also wanted to look at how these populations changed following treatment. So we had some post-treatment samples as well. And then in addition to the CyTOF, we also performed RNA sequencing and attack sequencing. And this was done also at Stanford, and the RNA sequencing was analyzed by the Dana-Farber Cancer Institute. So this is an example of the CyTOF results from the T cell panel. So CyTOF is cytometry by time of flight, which is, it's similar to flow cytometry if you're familiar with that, But instead of using antibodies that are conjugated to fluorophores, it uses antibodies that are conjugated to lanthanide metals. So you don't have the like compensation issues that you do with flow, and you can create much more robust panels that have a lot of markers. So at the time, this technology was much better than flow, just to look at large panels of markers. So here, in an example of baseline apheresis, you can see a representation of all these different immune cell types. You have your myeloid cells, your B cells, and your T cells. So this is just kind of how we define the populations. And one of the differences that we found in the baseline apheresis was in the CD8 T cell clusters. So CD8 T cells are the cytotoxic T cells, and they're the ones that can directly recognize and kill the tumor cells. So one thing that we found was that patients who had good CAR-T cell expansion had higher levels of these naive CD8 T cells. You can think of them as being like more fresh, like they haven't been activated. They're more they're younger. You can think of them as younger cells, so they do a little better. And then they also had good expanders had a reduction of terminal effector cells. So these are cells that may be closer to being towards the exhausted side and later on. So that was one big difference that we found. This was also confirmed by flow cytometry, how you have a reduction in naive T cells in this pore expander group. And then from the RNA sequencing, we saw some interesting signaling pathways that were enriched in good versus poor expanders. So here in the orange, you can see that patients who had poor CAR-T cell expansion were enriched in T regulatory gene signatures, as well as a Th2 type phenotype driven by IL-4, whereas the good expanders had much more enrichment in naive T cell signatures. So that kind of went hand in hand with seeing the increase in this naive CD8 cluster. And when we looked at the myeloid populations, it was really striking because we saw that the poor expanders had much higher levels of myeloid cells, in this case, just monocytes and dendritic cells. And this is mostly driven by the monocytes. So that's kind of what we focused on in subsequent analysis. This is a multi-dimensional scaling plot, which plots the similarities or differences between different samples. So the closer the samples are together, the more similar they are, the more spread out they are, the more different they are. And you can see that all these poor expanders are kind of grouping together, where there's a lot more diversity in the good expanders. So that suggests that there's some common signature in the poor expanders that may be limiting that CAR-T cell expansion. And we really wanted to focus on those monocyte populations because we didn't see differences in the dendritic cells. There's only 2 dendritic cell clusters here, but this group was just so dramatically different between the poor and good expanders. So these are sub-clustered, and this is really granular. You don't worry about reading all the markers, but the kind of 3 main buckets that we put the monocytes in are classical, intermediate, and non-classical. And then we also looked at their expression of CXCR3. So monocytes are typically thought of as moving from classical monocytes, which are defined as CD14 high, CD16 negative, through intermediate and then into non-classicals kind of as they mature. And these populations are not very well defined in cancer, but they're thought to have very diverse roles in terms of how they interplay with the immune system. So classical monocytes are thought to be more pro-tumorigenic, whereas the non-classical or patrolling monocytes have been shown to contribute to more anti-tumor functions. And that was work by our collaborator, Dr. Lynn Hedrick. So when we looked at the differences in these populations, we noticed some interesting changes. So in general, the non-classical monocytes, or these NMOs, were more associated with patients who had good CAR-T cell expansion. However, when you look at the classical monocyte population, the CXCR3 positive or high classical monocytes were associated with good expanders, but CXCR3 negative classical monocytes were associated with poor expanders. So we saw this interesting association with the CXCR3, that all the monocytes expressing CXCR3 were associated with good expansion. And then we also did some flow cytometry looking at kind of this more traditional myeloid-derived suppressor cell phenotype by flow, which is defined as lineage negative, HLA-DR low, CD33 positive, CD11b positive cells. And these were found to be much higher in the patients with poor expansion. And when we looked at the classical monocytes specifically, the CD14 positive cells, those had more of this MDSC phenotype as well. And we did a analysis to kind of see which of these markers were the most important. So this is a bioinformatics analysis called Random Forest that ranks all the markers in the CyTOF panel based on their importance in terms of how different they are between good and poor expanders. And the CXCR3 came out on top as the most, I don't want to say predictive, but most important marker that's differentiating these two groups. Also followed by CD33 and CD357, which is the GITR receptor. But it's all pointing to the CXCR3 expression. So it's just a really interesting phenotype. And as I'd mentioned, these monocytes, as they differentiate, they go from a classical to intermediate to non-classical phenotype. So through pseudo-time trajectory analysis, grouping these cells based on either samples from patients with good expansion versus poor expansion, we found that patients who had poor expansion really had this, it looked like what appears to be like a stalling in their differentiation, that you have much more of these classical monocytes and very few of these non-classicals. Whereas the good expanders, these cells are able to fully differentiate and go through to these non-classical phenotypes. And you can actually, we did a correlation analysis looking at the peak CAR-T cell expansion and found that the non-classical monocytes, specifically CXCR3 positive ones, were significantly positively correlated with CAR-T cell expansion, whereas the CXCR3 negative classical monocytes were significantly negatively correlated with CAR-T cell expansion. So we're currently looking at the role of CXCR3 and what this could be doing to the monocytes, whether it's just a marker of a particular monocyte phenotype or whether it's a driver of this function. And so we were also able to look at some of the patients post-treatment of CAR-T cells. So now we're, all the data I've showed up to this point was looking at baseline apheresis. And this is looking at 2 weeks after treatment with the CAR-T cells. So we see that after CAR-T cell treatment, the patients who had poor expansion actually had an increase of their absolute monocyte count in their peripheral blood. And they were associated with lower levels of GM-CSF and IL-12. And if we look at these classical monocyte populations, we see that at baseline, which is the data I was showing you before, the poor expanders have much higher levels of these classical monocytes. But after CAR-T cell expansion, now these good expanders, which had low levels, now look like the poor expanders. And this might explain or be related to the fact that these CAR-T cells didn't really persist. And then the inverse is true, the CXCR3 high positive population that was originally high in the good expanders is now reduced after CAR-T cell treatment. And then just looking at CXCR3 in general across all the populations, we see this reduction in the good expanders. So we're still kind of looking into this, and we don't know whether it's these monocytes are directly interacting with the CAR-T cells or whether they're just associated with their expansion and lack of persistence. And then Chrissy, the graduate student in our lab, took this data and went back to a TCGA data set. So looking at the target osteosarcoma data. So this is RNA sequencing from the primary tumors of osteosarcoma patients, and found that patients who had higher levels of CXCR3 had an overall better probability of survival compared to those that had low CXCR3 expression. So we think that this may be like biologically relevant into how these cells are responding to tumor and responding potentially to CAR-T cell therapy as well. So in summary, we found that patients who had good CAR-T cell expansion had increased naive T cells, specifically naive CD8 T cells, as well as the CXCR3 positive monocyte populations. And this was interesting to us because typically people think of myeloid cells as always being bad. Like you think of myeloid-derived suppressor cells, you think of myeloid cells damping down T cell responses. So it's really interesting that now we have this population that's actually associated with good expansion. And then inversely, patients that had poor expansion had increased terminal effector T cells, as well as the CXCR3 negative classical monocytes. And then I didn't go into this data today, but we also saw increases in exhausted CAR-T cells in the actual cell product in those patients. So overall, from these studies, we identified several differences in the immune cell populations between patients who had a robust CAR-T cell expansion versus those that did not. And this is informative because it could lead to the generation of biomarkers that could predict whether a patient could be a good candidate or not for CAR-T cell therapy, and can also identify potential targets to modulate the myeloid cells or other pathways to design new therapies to improve outcomes to CAR-T cell therapy in the future. So I think like understanding and ultimately improving how CAR-T cells expand in the patients is the first step to increasing outcomes for solid tumor patients, because although CAR-T cells have been affected in leukemia and lymphoma, solid tumors have been a lot harder to treat with CAR-T cells, and not and one of the big differences is that tumor microenvironment. But it's important to note that it's not only in the tumor, it has systemic effects on the immune system throughout the whole body. So understanding what's happening with other immune cell types and potentially targeting that could improve CAR-T cell therapy in the future. Currently there is we have a follow-up trial that's now open at the NCI as well as some other sites. So based on the first trial, learning that CAR-T cell product had an increased exhaustion signature, Crystal Makel's group has developed a new GD2 CAR, which has different signaling domains, but also improved manufacturing. And this is now in trial. It's the GD2 CAR-Persist trial, also in patients with osteosarcoma. So instead of the instead of the OX40, there's now a 4-1BB co-stimulation domain. It's a dose escalation trial as well, looking at 3 different dose levels. And one of the big improvements in the manufacturing of these CAR-T cells is the addition of dasatinib to the culture. So one of the issues that they found in the original CAR construct was that it was tonic signaling, meaning that the CAR itself was signaling even when it wasn't seeing its target. And that led to an exhaustion of the CAR-T cell. And you can think of it, if it's just going, it's going to get tired before it gets to where it needs to do its job. And it was just signaling in culture. But the addition of this dasatinib in the manufacturing process prevents that signaling of the CAR. So now when you're infusing these CARs into the patients, they're more fresh, they're more new. They haven't been signaling the entire time during the manufacturing process. So that has been a big improvement. The other improvement with this current trial is that in the first trial, we only used cyclophosphamide for the chemotherapy preconditioning regimen, whereas since then it's now well published that a combination of cyclophosphamide and fludarabine is a much better pretreatment and has better outcomes for the T cell expansion as well as patient outcomes. So now it's a Cy/Flu regimen. And excitingly, we're planning to do, and we've already started on the first few patients, to do the same or similar correlative studies on this new trial that we had on the first trial. So now we can compare these patient cohorts and see how these improvements to the manufacturing and the CAR and the treatment, how those improvements pan out in terms of impacts on the immune system and whether it correlates with hopefully responses. So I think it's a great example of going from the bedside back to the bench, understanding what's happening both in terms of the actual CAR product, but how the patient's responding to that product, and then using that to design the new wave of therapy and going from the bench back to the bedside. So again, I just want to point out this is a really big effort and a lot of people were involved through many institutions. So mainly, I want to shout out to the Ramakrishna lab at Stanford, Crystal Makel's lab, as well as the Hedrick lab, which is now at Augusta University. And of course, to the whole clinical team and the patients and their families that were involved with these efforts. So thank you, and I'm happy to take any questions.
Thanks so much, Dr. Kaczanowska. That was super interesting. And everyone, please feel free to add any of your questions to the Zoom Q&A, and we'll make sure we ask those. And I thought I might just start with a couple and kind of going back, I mean, so this study that you did was looking specifically at expansion, and it sounds like the next study, the PERSIST trial, is then kind of looking at persistence. And I was wanting, and maybe actually, if you don't mind flipping back, there was one of your earlier slides that showed all the patients on study and kind of time to progression. And
oh yeah, the swimmer's plot.
Yeah, and wondering, I mean, I know ultimately they all progressed, but some took a little bit longer.
Mm-hmm.
And wondering if there was any kind of correlation between expansion and time to progression.
Yeah, so I don't believe there was. And again, this is just a very small cohort of patients, but I don't think it correlated in this trial. But in general, expansion is the first step to response. Like, I don't think you could have a really robust response without expansion. But just because you have expansion, you may not get response as is evident in this trial. So I think step one is try to improve expansion, and then we may have to target other factors as well as maybe some of these other immune populations to see if we could improve the clinical response.
Yeah, likewise, I'm curious about just other factors that might affect expansion. So, for example, GD2 expression, if a patient has a lower or higher GD2 expression in their tumor cells, could that?
So in general, the GD2 expression is very robust. I don't know of a case where they've lost GD2 expression. In this trial, we did not go back and look at expression at the end. But I know in other cancer types, I was just at a seminar earlier today that Sneha was giving, and she's looking at diffuse midline glioma, and they looked at that and it does not lose GD2. I'm not familiar if somebody's done that specifically for osteo.
Yeah,
but we think it's a failure more so on the CAR-T cell level.
okay, so not necessarily any correlation, or maybe didn't correlation between level of GD2 expression versus expansion. Because I mean, I think I had heard like back, 3 or 4 years ago, like GD2 is basically on every osteo cell, and it's high, but I don't know if that's the case. I think there's probably been some more learning since that. So I wasn't sure if there's more of a range still, like even though most cells probably do express GD2, if there's a range and if there's any kind of correlation between expression level and expansion.
Yeah, so I'm sorry, we didn't look in this trial. I'd be curious to see if anybody specifically looked at that.
Yeah, and then the other
Yeah.
thing of interest too might be disease burden for CAR-T therapy in general, like whether or not if someone has, unresectable disease, if the level of disease burden has any correlation to expansion.
Yeah, so we were thinking along the same lines, and we did look at that. We did not find any correlation with disease burden on this trial. And Rosie, as well as Julia Siegel in our group, and a few others have been working on trying to develop a disease burden score specifically for metastatic solid tumor patients that's more representative of overall tumor burden and not just focusing on one lesion. So there's a little bit of that in this paper, kind of trying to define this new disease burden score. And I think they're still working on that, and also trying to use AI to help develop a better way to quantify disease burden in metastatic patients. But we did not find any correlation here with expansion and disease burden.
And what about so I know that there were a couple of patients that did experience the cytokine release syndrome, and wondering if is that like an indication of increased activity or expansion?
In general, yes. In this particular case, I don't believe so. I don't think it wasn't like the patients who had CRS had a higher expansion.
Okay.
It wasn't correlated here, but again, this is a very small patient size.
Right, but it was kind of because it was one of those, like outward things that you could, track. And like full disclosure, actually, Dylan, my son, participated in a GD2 CAR-T trial at Baylor, actually. And but I remember it was one of those things where you're like wanting to see some sort of outward sign that like something's happening. So I was just curious if that was an indication of any increased expansion activity.
I'd have to go back and check, but I don't believe, I think we looked at, I don't think that the patients that, because I think there were like two main patients that had really high levels. I don't think those were the two that had the longest disease. I can double check, but I don't believe that was the case.
Got it. And so I'm wondering, this is probably, I guess, something that you're studying in the PERSIST trial, but determinants of expansion, like how that correlates with persistence. And is that basically the goal of the study, is to determine if they're same or different?
Yeah, so in this trial, we didn't see persistence, but the hope is that with the new CAR-T cell construct and with the new manufacturing protocol, that we will see persistence in the new trial. So we're starting to analyze some of the data from the first few patients, so I can't really speak to that because it's all still very new. But the hope is that we will have better persistence. And you're correct, like if we see some patients that have more persistence versus others, then we can look and compare to see what other differences are present.
And then that was so interesting about let me see if I can get all the letters right. It was the CXCR3, I think.
Yeah,
that's what it was. Seeing that correlation, and as you mentioned, like potentially you might be able to define biomarkers that could identify good responders to CAR-T cell therapy. But I wonder, is there any implications from what you've learned here about when's the ideal point for the apheresis and the collection of the initial sample from which you're manufacturing the CAR-T cells? I mean, is there any implication, I guess, to kind of move the needle by kind of dialing, turning the dial on when you actually collect the sample?
I mean, I'm not a clinician, so I can't speak to kind of the logistics of that, but I think like once a patient's enrolled, I think they try to collect and make the cells and get on study as swiftly as possible. So I don't think they're delaying for any particular reason, maybe like a washout of previous therapies. But I don't think there's a specific kind of ideal day.
Yeah,
I don't know if people have looked at that.
I guess what I'm thinking about um, is, and I know this is difficult because I understand also that this is like a very expensive process, that actual manufacturing. So it's not
Hmm.
something that you can just do flippantly and just kind of be like, well, let's create CAR-T cells for everyone. But you know, often at times when patients are enrolling in a trial like this, they have likely gone through many rounds of treatment,
Hmm.
different cytotoxic drugs, and their immune systems are probably pretty tired. You can get those like tired senior T cells, um,
Yeah.
not the fresh ones. And so wondering like if and you know, this is kind of like in a dream world, but you know, if you were to be able to collect the sample up front at an earlier point prior to when the patient is really needing it or about, you know, to enroll, and basically just bank it, you know, so you're almost
Hmm.
like kind of collecting at an earlier time and kind of banking it for when they might need it at a later point, when their immune system might be stronger and perhaps like more of those naive fresh T cells. Is
Hmm.
that, I'm just like wondering if kind of the point of apheresis, if that's kind of a way to move the dial on having kind of more naive T cells.
Hmm. I mean, I would love to see that. I think at this stage in development, CAR T cells are not front line. So a lot of like all the patients coming in for these trials have already undergone multiple rounds of prior therapy. But I think ultimately, if this is shown to be effective, that it would move to more upfront therapy. And yeah, I would hope that if you have less rounds of chemo, less rounds of radiation, you may have a better starting cell product that may be more effective.
Yeah. I mean, I wonder, like, could you, I don't know how much you need to manufacture, but like even if you collected the, you know, the blood samples and froze them earlier in
Hmm.
treatment, and then like are you able to manufacture CAR-T cells off frozen product like that?
Hmm. Yeah, I believe they do. Um, I think the apheresis is collected. I'm not super familiar with the logistics of the manufacturing, whether they're frozen and then manufactured and then frozen again, or whether they're manufactured fresh. But in general, human T cells are able to undergo like freeze thaw and do relatively okay. So I'm not sure if somebody's looking at that, but it sounds like it would be a great idea.
Yeah. It's just it's kind of, you know, it's kind of those like overall odds that are sometimes stacked, I think, for on clinical trials because by the time
Yeah.
patients enroll, they're all, you know, it's like disease progression and aggressive disease. And you almost
Hmm.
wonder if you could try these experimental therapies at a more ideal time point when there's
Hmm.
no disease burden, low disease burden, or like a fresher um,
Hmm.
blood sample, whether that might just help with the efficacy. Um,
Yeah, I think it would because all that chemotherapy is definitely impacting your immune system.
yeah. Um, okay, so then I had another question about, because there are a couple of other GD2 CAR-T trials, like the one that I mentioned that was at Baylor, and I think
Hmm.
UNC has one. And um, and so a couple questions, I guess, from like a patient perspective, if you're shopping around for a GD2 CAR-T trial, kind of what the real differences are. Is it just like in how the CAR-T cells are manufactured? And then also just curious, like if there's some collective learning because across these GD2 CAR-T cell trials, that you know might just kind of
Hmm.
make all the learning a little bit more robust if we're kind of pulling information together from these different trials.
Yeah, so I want to start with a disclaimer that I am not a clinician. So I don't want to make any recommendations. But I do want to point out that the GD2 construct used in this trial was the same one from Baylor. And I think that in general, the pediatric immunotherapy space is pretty collaborative. And I know like the current PERSIST trial is multi-center. So it's at NCI, it's at Stanford, it's at CHOP. I'm not sure if there's other participating institutes as well, but I think that people do talk to each other and try to learn from other experiences, because these trials are very small, and you want to try to learn as much as you can from these patients. And I think that's also exemplified in the trial I presented today. Even though we did not see really robust clinical responses, we still wanted to learn from this trial and try to not, you know, to just try to like find something that could help improve the next iteration. So I think it's important, even if you don't get strong clinical responses, to still learn something from these patients.
Yeah, I mean, the information about the monocytes and the CX6R is really, I'll get those alphabet and numbers fraction one day.
Yeah, so, and we're trying to learn more about like whether that's an association or a driver and how that's related, and hopefully that could lead to either a biomarker or some other therapy that we could combine with CAR-T cell therapy. So that's the ultimate goal.
All right, and then we did have one question from the audience. Do you know what the minimum number of PBMCs is required to undergo a clinical CAR expansion, or are the T cells separated at the beginning?
Yes, so actually in this trial, the manufacturing process kind of switched halfway through. The first few patients, they did a CD3, CD28 expansion. So those are antibodies that provide signals for T cells to proliferate. So it like selects for the T cells in the culture. And then the last, I believe, 4 patients, their product was actually elutriated, which means that the apheresis went through this elutriation process that is, I believe, just based on like physical properties of the cells. So different immune cells can have different sizes, and they fall differently in a density gradient. So the elutriation would actually enrich for the T cells, and then they're cultured. So I think, and I'm not sure, I've like different, like you mentioned, Christina, different institutes may have different ways that they manufacture and prep their cells. But I think that can make a difference. And I know people are looking to see the best way to do that. In terms of cell numbers, I really can't say. I don't know how many cells you would need to make a product. But the good thing about the T cells is they do expand in culture. So you end up with much more than you started with.
You know, I'm wondering if the expansion, because they're kind of like all these, you know, different dials, right? There's like, how do you improve expansion? How do you improve persistence? And even like the whole lymphodepletion is like a way to kind of improve expansion. But I'm wondering if it's like expansion kind of like could you solve the persistence issue by just continuing to to infuse, like to continue infusing at different points? And so is expansion maybe kind of more critical versus persistence, because you can maybe solve persistence with just continue to administer?
Yeah, I wish there was an easy answer. Unfortunately, it's not that simple. I don't know if actually, yeah, I can't speak to the GD2 CAR specifically, but in general, in the CAR T cell field, I know some groups have re-infused. To my understanding, it's usually not the home run that you would hope it would be. And then there's questions whether you retreat with the chemotherapy preconditioning for a second infusion. But in my understanding, I don't think a second infusion has been super successful. And I think it's also probably a case by case basis, and some of the mechanisms why the first infusion failed, whether it's that the CAR T cell itself didn't persist, whether you lost your antigen, which can be the case in some of the like lymphomas or leukemias. So there can be a multitude of reasons the CAR T cell failed. And unfortunately, it's not as easy to just give a second dose. A lot of times you don't get as robust expansion on that second dose. It's not been super effective to my understanding.
All right. This was so interesting. I really appreciate your taking the time to join us today, Dr. Kaczanowska. And like you said, team effort, really great work. And we're going to, I put links to your full paper, as well as the 2 trials that Dr. Kaczanowska mentioned in the chat. And we'll also put those in our follow-up email with the recording of the episode. So you can kind of look at the info in more detail, but so interesting. And thank you for taking the time to share the learning from that study. Really appreciate it, and appreciate all you do to make it better for pediatric sarcoma patients. More information on this and all our OsteoBites can be found on our MIB Agents website at mibagents.org and on our YouTube channel, and also at your favorite podcast place. And we hope you can join us next Thursday on August 8th. We're going to be joined by Dr. Diana Azam from Florida International University. She's going to be discussing the clinical utility of functional precision medicine to guide treatments for osteosarcoma patients. And her presentation will be focused on describing the foundational principles of functional precision medicine as applied to pediatric cancers, and how major technological improvements, as well as an explosive broadening of the cancer pharmacopoeia, has enabled the bridging of genomics with functional drug testing as clinical decision support tools to impact patient care. You can find our OsteoBites lineup for the next few months on our website. And if you ever have any ideas of topics you'd like to hear about, please feel free to email us at events@mibagents.org. Thank you again, Dr. Kaczanowska and Andrew, for spending an hour with us today. And thanks to all of you for taking time out of your busy days to join us. We hope to see you back here next week on August 8th when we talk to Dr. Azam.